Hi everybody, my name is Shrushti and today we are going to discuss the physiology of memory in this video. We all know what memory is, but the fascinating fact is that how are we able to store and recall so much amount of information which is stored in a very small part of our body, that is our brain. Have I seen this person somewhere? Why is this question coming to our mind always whenever we see a familiar face? Why is the earth round? We never draw it as a triangle or as a rectangle. Why do we always color a banana yellow? It implies that all this kind of information is stored in the brain in this prefrontal area of the brain which is marked in yellow. It is divided into various compartments which stores information regarding shapes, sizes, colors, actions, etc. And a combination of all this information will help you to prognosticate, that is plan for a future event, delay our action to an incoming signal. For example, we don't hit our mom when she gives us a slap, which implies memory is also stored on the basis of moral values. Apart from this, it can be used to solve complicated mathematical problems, diagnose diseases, etc. So how do we generate this memory? The thought process plays a very important role in generation of memory. The input to the brain, anything that comes to our mind is supposed to be the input to which a thought is generated. This thought is acted upon various systems of the body, that is the limbic system, which takes care of the emotional aspect of the brain, the cerebral cortex, which takes care of the practical aspect of the brain, the thalamus, the reticular formation of the brain, and many other systems. And all this together will result in the formation of a conclusion, which is the output, and this output will finally determine our action to this incoming input. So the question is, how do we remember things? If you look at the neurons here in this picture, the neurons are just like these confusing roads. When there is a change in synaptic transmission, that is basic change in the sensitivity of the neurons will form pathways like this 8-lane roads, and these pathways are known as memory traces. For example, we must have experienced that when we read a concept for the second or the third time, it is much easier and it is less time consuming because these memory traces are already formed for that concept. So the question is, how do we decide that which part of information is required to be stored in the brain? This brings in the concept of sensitization and habituation of the synaptic transmission. Habituation is a kind of negative memory. For example, if you keep listening to the same alarm ring every day in the morning, we stop responding to it, which implies the brain is habituated. That is, the brain is used to that kind of memory, and therefore this kind of memory is discarded by the brain. The second type is the sensitization, which is a positive type of memory. Any kind of thought which gives you pleasure, for example, a date with your loved one, or the thought gives you pain, for example, your accident. This kind of memory will be very well stored in the brain. That is, the brain will be sensitized to this kind of memory, and therefore it is supposed to be a positive kind of memory. The best part is the classification of memory. Classification can be on various types. The first is on the basis of duration. It can be divided into short-term memory, intermediate long-term memory, and long-term memory, which can be depicted in this picture. The other classification is on the basis of memory stored, which is declarative memory and skill memory. Declarative memory is a mere recollection of facts. For example, when you were out on your first date, you very well remember the color of the shirt you were wearing, the food you ate, the song you played, the atmosphere, etc. There is no but or it to this information because these are mere facts and they are saved as it is in your brain. The next one is skill memory. It aids in the performance of a particular type of task without conscious awareness of previous experiences. For example, driving a car is supposed to be a skill, and after you learn this skill, you can drive your car at a subconscious level. That is, we can take care of all the functions, that is the steering, the accelerator, the brakes, and other functions at a subconscious level. Let's start with short-term memory. This kind of memory lasts only for a few seconds to few minutes in a brain. This theory is explained by temporary reverberating circuit. That is, the neurons are going to form circuits like this, shown in the picture. Only till the time you keep thinking about that information will that information be stored in your brain. For example, if you try to remember a phone number which has around say 10 digits, only till the time you keep thinking and reiterating these numbers in your mind, you will remember them, and once you stop it, that is the neurons stop going through that circuit again and again, this kind of memory is going to get vanished from your brain. This is the concept of short-term memory. The next and the most interesting part is the intermediate long-term memory. This kind of memory can last for minutes and even for weeks. The memory traces have to be activated enough to make them permanent. That is, small amount of rehearsal is required to make such kinds of memories. These types of memories result from temporary chemical or physical changes. I repeat, temporary chemical or physical changes are going to occur in the synapse, which will in either the presynaptic terminal or the synapse postsynaptic terminal, that is either prior to the synapse or after the synapse. This mechanism of memory was very well studied by Kendall and his colleagues in a large snail aplysia. To explain this theory, let's consider three neurons: the sensory terminal, the nerve to be stimulated, which are directly connected, and the facilitator terminal, which is directly connected to the sensory terminal. Let's talk about a normal stimulus which is given to the sensory terminal and because of which the nerve to be stimulated gets stimulated. Now, if you keep giving the same stimulus to the sensory terminal, a time is going to come when this sensory terminal is going to get habituated to this stimuli, and therefore the signal transmissions to the nerve to be stimulated will become weaker and weaker and finally disappear. For example, let's take the same example of the morning alarm. If you keep listening to the same ring every day, you will stop responding to it. At the same time, if you stimulate the facilitator terminal with a noxious stimuli, if you give a noxious stimuli here, which is supposed to be a painful stimulus, the signal transduction to the nerve to be stimulated increases. For example, along with the morning alarm ringtone, that is the stimuli to the sensory terminal, if you give a noxious stimulus, that is a mother's tight slap to the baby, the signal transmitted to the nerve to be stimulated will become stronger and stronger, and it will result in a quicker action, that is, you get up suddenly. Which implies even after habituation has occurred of the sensory terminal, this pathway can be converted back into a facilitated pathway with only a few noxious stimuli. So let's discuss how does this happen. So for this we are going to talk about the molecular mechanism of intermediate long-term memory. First, let's discuss about the mechanism of habituation. For the habituation of the sensory neuron, we require it to stop releasing neurotransmitter to the nerve to be stimulated. For this, whenever habituation occurs, there is progressive closure of the calcium ions in the sensory terminal. Because of this, much smaller amounts of calcium is released into the sensory terminal. Therefore, decreased amount of neurotransmitter is released, because we all know that calcium is very important for the release of neurotransmitter from the neuron. As a result, decreased stimuli goes to the next neuron, and therefore the next neuron doesn't get stimulated. The mechanism of how there is progressive closure of the calcium channels is still not known. The second is mechanism of facilitation. So when there is simultaneous stimulation of the facilitator and the sensory terminal, it results in release of serotonin at the sensory terminal. This serotonin is going to act on the serotonin receptors present on the sensory terminal. The serotonin receptors will activate this enzyme, adenyl cyclase enzyme, which is going to result in formation of cyclic AMP, that is cyclic adenosine monophosphate. This cyclic AMP activates a protein kinase, which in turn causes the phosphorylation or the activation of another protein, which causes blockage of potassium channels. Due to lack of potassium conductance, it causes greatly prolonged action potential in the sensory neuron, because we know that only when the potassium is going to go out of the neuron will get repolarized. So since the potassium is not going out, the action potential will prolong for a very long time. Till the amount the action potential is prolonging, the calcium channels are going to work, which implies great amount of calcium ions are now going to enter into the neurons, which ultimately will release in greater amount of neurotransmitter release, which in turn will stimulate the nerve to be stimulated at a greater rate. This explains the mechanism of facilitation. In the end, let's talk about the long-term memory. This type of memory is believed to occur because of actual structural changes at the synapses. I repeat, actual structural changes are going to occur at the synapse, and this is going to either enhance or suppress signal transduction. What kind of changes are these? For example, there can be an increase in the release site of secretion of transmitter substance. That is, we can increase the surface area to which the vesicles are going to fuse, or we can increase in the number of transmitter vesicles, or we can increase the number of presynaptic terminals. That is, instead of a single sensory terminal stimulating the nerve to be stimulated, we can take 3 to 4 sensory terminal which will stimulate the same nerve to be stimulated, or we can make the dendrites of the nerve to be stimulated very strong so that they transmit more stronger signals, which implies development of a true long-term memory depends on physically restructuring of the synapse themselves in a way that change their sensitivity to the transmitting nerve signals. Let's discuss the concept of use it or lose it. It means that if you don't use the brain, your brain will never develop. This concept governs the final number of neurons and their connectivity in respective parts of human nervous system. For example, that means that the neuron which gets stimulated, the connections which this neurons make with the other neurons will decide the memory traces which are formed. That is, only when you use the neurons will the neurons get developed and will connect to the other portions of the brain. For example, if you block one eye of an infant since their birth, the memory trace from the retina to the visual cortex will not develop. As a result, when this cap is removed, the child will be blind from the respective eye, and the number of neuronal connections is determined by the specific nerve growth factors released, because we know that nerves grow only on the basis of nerve growth factor released. Next is consolidation of memory. It means the conversion of a short-term memory into a long-term memory. So this happens by a short-term memory being converted into temporary physical and chemical changes. After that, anatomical changes are going to occur, and finally a long-term memory is going to generate. Minimal consolidation requires only 5 to 10 minutes, but for a strong consolidation we require 1 hour or more. Rehearsal enhances the transference of short-term memory into a long-term memory. Hippocampus is the region in the brain which is going to promote consolidation. Convulsions, sudden application of very deep anesthetics can prevent this kind of consolidation. Codification during consolidation. Codification implies to the sorting of the memory. For example, like we always keep the t-shirts together in the closet, like shown here, all the footwear together, all the dresses together, or if you take the example of a bookshelf, we keep all the anatomy books together, all the physiology books together, all the biochemistry books together. It becomes very easy to store it and to search it in the near future. It implies codification means similar types of information are pulled from the memory storage bins and are used to process the new information. That is, the old and the new are compared for similarities and differences, and they are stored processed and not stored unprocessed. And this is necessary to be able to search the memory store at a later date to find the required information. Let's talk about the role of hippocampus in consolidation. How does the hippocampus get to know that which kind of memory is to be stored and which kind of memory is not to be stored? So any kind of thought which gives you a reward or which gives you a punishment is always stored in the brain. Reward centers involve stimuli such as pleasure, happiness, excitement, etc. For example, if you have topped a university exam, this kind of stimuli is going to give you pleasure, happiness, excitement. That is, it is going to give you a sense of reward, and therefore the brain is going to store it inside. With respect to the punishment center, it involves sensory stimuli like depression, anxiety, pain, etc. So anything in your life which is giving you any of these things is stored in your brain, so that next time when you try to repeat the same thing, it tells you that this kind of thing is going to give you a punishment. So therefore, we store information regarding the reward and the punishments we get from that particular stimuli. In the end, let's talk about the most interesting part, that is the diseases associated with memory. The first disease is anterograde amnesia. It is the inability to establish new long-term memories. That means the person from now on cannot establish new long-term memories. It occurs due to lesions in hippocampus or surgical removal of hippocampus, because this hippocampus is required for the consolidation of memory. Such people cannot store verbal and symbolic types of memory, because these types of memory, they require processing, and this processing is done in the hippocampus. So therefore, they are not able to form long-term memories and not even intermediate long-term memories which last for a longer time. These people are unable to establish new long-term memories on those types of information that actually form the basis of intelligence of a person. The next one is retrograde amnesia, which is just the opposite to anterograde amnesia. It is the inability of the person to recall memories from the past, which implies from now on the person will not be able to recall memories which are stored in the past. The degree of amnesia for recent events is likely to be much greater than for the events of distant past. That is, the memories which are of recent past will be more under amnesia, that is, they will undergo more of dementia as compared to the memories which are of distant past, because the memories of distant past are till now stored into a very deeper gradient of the brain. Damage in the thalamic area of the brain specifically leads to retrograde type of amnesia, because thalamus plays a very important role for a person to search the memory storehouse and thus read out the memories. So, as I told you before, the memory process not only requires the storing of memories but also an ability to search and find the memory at a later date. Hippocampus is not important in reflexive learning. So, people with hippocampal lesions can learn certain types of physical or skilled movements which are known as reflexive or skill learning. People with hippocampal lesions will have difficulty in learning information which involves symbolization or verbalization, but not the information which involves physical or skilled movements like swimming, skating or any kind of sport. Let's take the example of this movie, Lafange Parinde, in which Deepika Padukone was blind but she could still excel in skating, because here since she was blind, she could not take care of the symbolic information, but skilled movements were not taken care of hippocampus and therefore she could excel in it. The last and the final disease and the most interesting part is the Alzheimer disease, which is nothing but the premature aging of the brain, usually beginning in mid-adult life and progressing rapidly to extreme loss of mental powers. If you can see in this picture, an eraser is used to erase the memory from the brain. It is usually a type of dementia, that is a loss of memory, which is very common among the old people. It is the loss of neurons in the part of the limbic system that drives the memory process, because we discussed the important part of limbic system in the development of memory. So, what are the clinical features associated with Alzheimer's disease? There is an amnesic type of memory impairment, there is deterioration of language, visuospatial deficits occur, that is, the patient is not able to judge the spatial orientation between different objects. Gait disturbances, motor and sensory abnormalities, seizures, that is epileptic attacks, which are generally uncommon, neuropsychiatric symptoms and behavioral changes which generally occur in the later stages of the disease. The causes of this disease, the most common cause is the accumulation of beta amyloid peptide. Now, what results in the accumulation of beta amyloid peptide can be mutations which result in the increased production of this beta amyloid peptide, Down syndrome, that is the 21st chromosome trisomy, abnormal genes that control apolipoprotein E. Apolipoprotein is a transporter protein which helps in transportation of cholesterol to the tissues. So, problem in any of these three can result in the increased accumulation of beta amyloid peptide, which in turn can be a result of Alzheimer's disease. The second one can be cerebrovascular diseases like hypertension, that is increased blood pressure, or atherosclerosis. And the risk factors to this atherosclerosis can be hypertension, diabetes, hyperlipidemia, etc. If you can see in this image, let's talk about the healthy brain. This healthy brain has got very prominent gyri and sulci. In mild Alzheimer's disease, this gyri and sulci have become very less prominent, and in severe Alzheimer's disease, they have decreased much less. So, this picture very well depicts the deterioration of the brain. So, I hope you enjoyed this video. Thank you so much for listening. If you enjoyed it, please hit the like button and the subscribe button, and for any queries, feel free to comment below. Thank you.